Hello everyone, of course. I'm Numair Masud. I must begin by saying that it's a great pleasure to be here today and really have an opportunity to discuss my PhD and all the research associated with it, and of course its implications. I'm extremely fortunate to be in the final year of my PhD that just involves a lot of computer work, and I don't have any experimental work to do. And I fully appreciate that there are a lot of people who have been disrupted by the global pandemic in terms of their research. So I know I am extremely fortunate, which is something not shared by everyone. And I know we're all living through uncertain times, and I do really hope that you're all keeping well. So I have titled my talk, Fishy Business, How Host-Pathogen Interactions Are Being Impacted by Global Change, quite literally because my entire PhD has utilized fish models to understand how various human-induced and natural changes impact host-pathogen interactions. The core of my research is inspired by one of the most profound concepts in life sciences, I believe, the Red Queen hypothesis. This is encapsulated by the drawing that you see on the bottom left of the first slide. Here we see Alice from Wonderland being speedily escorted across a landscape, and she notices that despite their extreme speed, they don't seem to be moving at all. And to this, the Red Queen responds, now here you see, it takes all the running you can do to keep in the same place. This quote embodies the host-parasite arms race that is integral to global ecosystems. For this talk, I'm defining a parasite as any organism that is metabolically dependent on another and causes some degree of harm to the host. It also helps address why research such as mine matters. Why should we care about how anthropogenic and ecological stresses impact these host-parasite interactions? Despite the yuck factor that parasites may induce, parasitic pathogens form the dominant biomass of all ecosystems. I really like referring to parasites as the dark matter of ecosystems, unseen but certainly not unimportant. Indeed, parasites influence all aspects of organisms' lives, from growth, mate choice, and if left untreated, mortality. Even from humanity's perspective, they have important economic and social impacts, from placing pressures on our food industries to burdening our healthcare systems. More worryingly, we are living through times of unprecedented global changes, where anthropogenic activities are stressing ecosystems and their organisms. With host-parasite interactions being so omnipresent, the question I'm interested in answering as a PhD student is, how do individuals and ecosystems respond to these stressors in relation to disease resistance? Broadly speaking, to address this question, my research utilized model host-parasite systems, the hosts being fish and their parasites. Fundamentally, the technique I have used involved exposing the host to specific stressors for a defined period of time, after which we experimentally infect hosts with parasites. Control hosts are, of course, not exposed to the stressor, but are also experimentally infected. We then monitor infection trajectories over time, which just means that we can non-invasively count the number of parasites. This allows us to understand how host disease resistance responds to stressors. Typically, for most transmissible infections, we utilize the SRI model, where hosts are either susceptible and their parasite numbers consistently increased, as shown by the red curve over here on the right, or are able to recover, as shown by the blue curve, where parasite numbers increase, followed by a consistent decline indicative of an immune response. Or hosts can, of course, be immune, where parasite numbers fail to cross a specific threshold, as shown by the green curve. Briefly here, I would like to outline three of arguably my favorite experiments that I've conducted on globally important stressors and their impact on disease resistance. The first graph on the far left is the result from an experiment that investigated how acute and chronic noise exposure affected disease resistance. Our results showed that fish experiencing acute noise, that is 24 hours of noise exposure, suffered the greatest parasite burden, as seen in the blue curve over here. Interestingly, host experiencing chronic noise, that is for 7 days, had the lowest parasite burden, as shown in the gray curve, But these fish died significantly earlier compared to those from all other treatments, demonstrating a functional trade-off between increased disease resistance and shorter lifespans. Now, often when we think of stress related to climatic changes, we neglect certain factors such as access to food that species would need in order to maintain a functional immune system. Now, if you focus on the middle graph, our investigations have shown that when fish are exposed to a more detrital-based diet, which is what they would be exposed to in winter times, their immune response to parasitic infections is much poorer, as represented by a higher mean parasite intensity, which is just basically more parasites over time, compared to those that are exposed to more invertebrate-rich protein diet, which they would be exposed to during the summer. Of course, in the real world, whether in managed or natural ecosystems, species are exposed to multiple stressors, not just individual ones in isolation that we can do in the laboratory. So my final PhD experiment was a multi-stressor one. Now, multi-stressor experiments are notoriously difficult to model due to interaction effects. So when a stressor works in conjunction, their impacts, as revealed by the hypothetical graph shown on the far right, could be either additive, where the combined effect of individual stressors equals the sum of the expected increased disease burden compared to controls, or less than expected, which is antagonistic, or more than expected, which is synergistic. For our multi-stressor experiment, we are using glyphosate and microplastics. Now, glyphosate is the most used herbicide globally and is toxic to aquatic organisms, and very, very controversial, certainly in the US. And of course, microplastics, as I'm sure many of you are aware, are found everywhere and are notorious for absorbing organic pollutants, such as glyphosate. We are still in the middle of analyzing this data, and so hopefully the next time I were to give such a talk, something will be hot off the press. I'm aware that I only have a few minutes left, so to wrap up my talk, I would just like to remind everyone why researching host-parasite interactions in response to stressors is so important. Now, looking at this from the human side of things, arguably the biggest challenge parasites pose for us in the long term is their significant impact on food supplies. In aquaculture alone, for example, loss to parasites is greater than $6 billion annually. And now with human demand for food increasing dramatically as the population expands, we are placing unprecedented pressures on our animal stocks, and this will impact their abilities to resist pathogens. From an ecological side, we have already seen how parasitic pathogens impact all major aspects of an organism's life, from growth all the way to mortality. Now more than ever, ecological parasitologists are beginning to appreciate just how the resilience of an ecosystem is dependent on healthy host-pathogen interactions. We're all living through a global pandemic that originated as a result of host switching that occurred when humanity decided to disrupt tightly co-evolved host pathogen systems by moving hosts to novel environments. It is becoming increasingly clear that we are creating new niches for parasites. I would just like to end on a more positive note by thanking my incredibly supportive supervisory team, Joe Cable and Amy Ellison, shown on the top left and top right, Joe Jackson, my collaborator, shown here at the bottom left in Manchester, who worked with me on the seasonal diet experiment, and of course my master student, shown here on the bottom right, Laura Hayes, who worked with me on the noise pollution project. And of course, thank you all for listening, and I would love to take any questions that you may have for me. Thank you very much indeed.
Fantastic, thanks so much, Numair. That was a really captivating talk, which I expect nothing less than from you. Does anyone have any questions? I can kick things off with one from me then. So obviously you mentioned about the healthy host-parasite relationships in ecosystems and how this sort of evolutionary arms race needs to stabilize. And there's obviously a trade-off for the parasites becoming too virulent and damaging the host. I've seen in several different examples. I imagine as well, there's a big consensus, particularly in agriculture and aquaculture, to try and get rid of these parasites. But I imagine from a sort of conservationist angle, maybe we don't want to eradicate the parasites. Maybe we want to keep some of them.
Exactly.
wondered where sort of parasitologists would stand on that.
I think they would certainly agree with you. I think now more than ever we understand that a healthy ecosystem is one that has a lot of parasites. I think sort of linked to Christina's talk about a healthy gut microbiota. I'm not suggesting that the microorganisms in our gut are parasites. I'm sure we can stretch the definition to include that. But as we need a healthy gut microbiota, so we need a healthy parasite system in any given ecosystem. Now, I don't know what that would look like, and I would argue no parasitologist would know what an ideal parasite ecosystem looks like, but certainly enough research has been done to show that in disrupted ecosystems, whether it's disrupted in the sense of there being a lot of microplastic contamination or a lot of persistent organic pollutant contamination, when you do a parasite survey, you can clearly see that in disrupted ecosystems, you do see parasite populations to be quite low, whereas in a healthy, quote unquote, pristine ecosystems, if you can argue such exist, there is a healthier abundance of parasites, or if you would like to say, just a greater proportion of parasites to hosts.
Fantastic answer. Thank you very much. Any other questions?
Yeah, I've got a quick question, if that's all right.
Yeah. Hey, Borja.
Hey, man. Your model for your fish model regarding the anthropogenic noise, is that been shown to happen in other species models, or is that specifically aquatic species?
So when you mean the effect that I've shown in terms of disease resistance, or in terms of noise being bad in general for the welfare?
Yeah, the latter, noise being bad in general for the fish and. Yeah.
So there are studies that have shown certain frequencies of noise don't seem to have any effect on fish. And then there are other studies that have shown that depending upon what factor it is you're focusing on welfare-wise, it can be quite bad. So for instance, there are quite a few behavioral studies that focus on anti-predator behavior that show, for instance, that in the acute phase, so in the short term, it's bad for anti-predator evasion, but then fish can seem to adapt to it over time. So I guess in this instance, this sort of temporal variation does make a difference. Then there are other studies that show when it comes to fish fry, acute noise and chronic noise is bad, full stop, and that it reduces the chances of survival all the way to adulthood. So I'm not really sure if that's an answer to your question, but it's not a simple answer to the question, because I guess it does depend on which welfare factor you're focusing on.
Yeah, that makes sense. Thank you.